Thank you. Good afternoon, good morning, good evening, everyone, depending on where you're located. I am Dr. Tia Warrick. I am the Public Health Director and Assistant Professor of Biology at Juniata College. We are located in Huntington, PA, which is just central PA, Pennsylvania, in the United States. And the research, as stated before, that I will be presenting is work that I've done with some of my students who have now gone on to do graduate work at other institutions, but we were able to work on big data for the state of Pennsylvania to look at the association of diabetes outcome and atrazine exposure among Pennsylvania counties. It's really important. I'm an environmental epidemiologist, so we do look at, and I do focus on specific chemicals in the environment and how that interacts at a biological, a genomic level, and then also taking into account the social determinants of health. So before we even go into the data and what we did for this project, just making sure we have a baseline understanding of type 2 diabetes. Usually in normally for a person who does not have diabetes, the pancreas produces insulin. And when it produces insulin, that insulin helps with the uptake of glucose into the cells. And as we know, glucose is sugars that is used to metabolize energy for our cells to process. In a diabetic patient, however, the pancreas produces less insulin. So if there's less insulin, that means less glucose is taking any uptake into those cells. So that means the glucose is hanging out in the blood and it's raising those sugar levels. That can cause a lot of different irreparable damage and side effects to patients who have diabetes. Atrazine is a specific chemical that we focused on. There are a lot of different chemicals that we can see in the environment. Unfortunately, polyaromatic hydrocarbons, polybiphenyls, we have all different kinds of metals, toxic lead that's in the environment. But for this particular study, we focused on atrazine levels specifically. So the reason why is because Pennsylvania, United States, is very rural. There are maybe 3 major metropolitan cities, and the rest is rural, like I said before. And so there's a lot of farmland, and atrazine is a common weed control. It's a pesticide used to kill weeds as part of the agricultural practice. Now, the pesticide, when sprayed into the soil, can leach into other areas of the soil and get into riverways, get into drains. And it can impact a lot of different sectors of wildlife, such as algae, plants, mollusks, and different types of fish and amphibians. And so there's been a lot of research on how it impacts reproducibility as well as behavior, and it can affect development. So I want to understand, as part of this One Health thought, that we're moving towards in public health, how is it impacting humans? So diabetes is a growing epidemic that affects 38 million Americans, many unaware of their diagnosis. And type 2 diabetes, once rare in children, is now rising due to childhood obesity and due to globalization, not just in the United States, but worldwide. We're seeing an uptick of chronic disease as far as diabetes goes. We're thinking about atrazine. Like I said before, it's a widely used herbicide persistently in the environment. It's used worldwide, not just in the United States. And there has been one documented study to show a farmer in China having gestational diabetes, and it was linked to endocrine disruption. So there is one study out there that shows some type of exposure to atrazine. However, the results are inconclusive because the farmer was also pregnant. So that also could have been a contributing factor to the gestational diabetes. Whether or not the exposure to atrazine exacerbated that outcome is still very unclear. So that makes the research question we're looking into even more apparent to get clarity. We're looking at the diagnosed diabetes of total adults 18 years or older, and we're doing the age-adjusted percentage in Pennsylvania. As you can see, it's essentially rising, right? Over the last 20 years, it's increased, and this is just looking at it from a national, state level, but globally, the trend is pretty similar, that the prevalence of diabetes is slowly increasing with each passing year. This right here is the state of Pennsylvania within the United States. And we're looking at the percent of people ever told they had diabetes, so diagnosed by a physician. And the darker the particular county, so this entire thing is just a state. This is not the United States. This whole thing is just the state of Pennsylvania. The US is huge. But within this particular state of Pennsylvania, there are over 50 counties. And so the darker areas are the counties that has the highest prevalence of diabetes, whereas the lighter areas has the lower prevalence of diabetes. We also looked at the geospatial distribution of the socioeconomic and demographic vulnerability index. Essentially, we take into account salary, home, income. We take into account insurance. We take into account transportation needs, all of those things that put you at disproportionately a higher risk for having a adverse health outcome. We created this index, which is also can be found nationally on the CDC website. But we looked at, again, the higher the numbers, the darker the squares are across the counties in Pennsylvania. And we saw that, similarly in the areas that had the darker blue, also had the darker blue, which is the higher prevalence of diabetes, also had darker percentiles for a higher social, economic, and demographic vulnerability index. Again, this is circumstantial, right? This is just geospatially looking at it. We didn't actually do any stats just yet to really see if there is a true correlation there. Another aspect that we looked at, not just the the geospatial distribution of the social determinants of health and diabetes, we also wanted to look at the distribution of atrazine, right? Because we're looking at that association between diabetes and atrazine while accounting for the social determinants of health. So we looked at the atrazine levels that was collected by the EPA from 2001 to 2018. So the last 17 or so years, we've collected the maximum people that were exposed across all of the counties to atrazine levels. And so what we've seen is that the atrazine levels, for the most part, were between 0 and 1 micrograms per liter. However, that again is safe according to the EPA. The EPA says that as long as it's below 3 micrograms per liter, it's fine. Right. However, there's no concept of time. As an epidemiologist, as we know, time is very important. So that temporality of looking at that constant exposure between 0 and 1 microgram per liter for the last 17 years in these particular counties, and the people that were are native to the state of the counties of Pennsylvania, has lived there that entire time, has been constantly exposed to this atrazine. So they when they state that it needs to be, below 3 micrograms, they mean in that single instance. They have not accounted for temporality. So this study really focuses on looking at that constant exposure for over the last 17 years. So like I stated before, the study focus was investigating the association between atrazine exposure and diabetes incidence in Pennsylvania between the years of 2011 to 2019. Our hypothesis were to look at the counties with higher atrazine levels will show increased diabetes rates, right? That's what we're hypothesizing to see, especially the counties that is socioeconomically vulnerable. We looked at environmental and socioeconomic factors. We explored factors like education, income, and physical activity, all the things that can also impact diabetes. We did a comprehensive look at both social determinants and environmental risks. And we want to know if atrazine is contributing to the diabetes epidemic, right? When we think of diabetes and most chronic diseases, it's been very well studied, thinking about eating well, thinking about exercising, access to healthcare. But, there's a lot of things when we think about more than just access. Those who live in poorer communities or less, I would like to say, flourishing communities, they are more likely to be disproportionately exposed to chemicals due to lax policies and zoning laws, such as brownstones and super, super fields where you essentially have a huge dumping of chemical waste that they cover up with dirt just a few inches below. So it's really important to kind of see how the leaching of those particular chemicals in these communities are being affected. So when we think about methods, how we uncover the atrazine and diabetes link, we looked at 67 counties and our sample size was 405. When we're thinking about 405, we're thinking about 405 populations because this is aggregate data across the 67 counties. And then, like I said, we explored looking at all the different social determinants of health of what can actually increase the risk of diabetes so that we can ensure that we're accounting for that. So when we do establish that correlation or association between diabetes and atrazine, we can be more confident in that association because we've accounted for other things that could be modifying that relationship between the two. So our data source was the Pennsylvania Behavioral Risk Factor Surveillance System, or the BRFSS. They had data on diabetes prevalence, body mass index, physical activity, fruit and vegetable consumption. We also used the United States Census Bureau, which had information on the socioeconomic variables like income, education, population size, and racial demographics. And then for the atrazine exposure data, we received that from the PA Department of Health through the data exchange. And we were able to get water contamination reports measuring atrazine levels in public water systems. And then atrazine levels characterized in bins based on concentration in water sources. So when we look at the eligibility criteria, what we included in the study, we only included counties that had complete atrazine data and diabetes incidence records, right? They had to have both because that's the association we're trying to find between 2011 and 2019. The data was binned into 3-year intervals for better statistical consistency because, again, this is aggregate data. So this is essentially an ecological study, right? We can't, we want to reduce the aspect of ecological fallacy, which just means that you cannot make aggregate data take those particular inferences, you make it an aggregate level, and then make conclusions about individuals, right? So we wanted to ensure that because the data is aggregate, that we are including larger bin sizes, because if we then create smaller bin sizes, we're essentially breaking up the sample size, and then we're reducing the power effect to actually see any type of statistical association if there is any. The counties with missing data on risk factors have undergone we did statistical imputation. So if there were some missing data, we did imputation by finding medians for those missing values so that it wouldn't be affected by outliers or extreme values. And then we looked at, for the exclusion, the counties with incomplete or missing data that didn't have, that we just couldn't imputate, wasn't really good. We did sensitivity analysis. We just excluded those extreme outliers that, wasn't accurate. We, again, tested it in the model and then removed them if they violated what the assumptions are. So for the methods of how we uncovered the atrazine and diabetes link, we did a multilinear regression. So we used this regression to predict the relationship between diabetes incidence, which is the continuous outcome, and predictors like atrazine levels, which is a continuous variable. We looked for ideal for handling multiple confounding variables like age, income, race, and BMI. So we had to look at VIF, which is essentially shows how correlated two values are together. And if these variables are really correlated, then we would figure out which one is the best one to keep in the model so that we are not essentially mediating an effect between the association of atrazine and diabetes. And so we essentially chose the best variables to keep in the model through the testing. So we originally had 536, but then once missing values were completed, 405 data points were analyzed. Minor imputations, and then we looked at it over the 9 years. Like I said, we tested for VIF. We also tested for normality and homoscedasticity. These are all the assumptions when you're doing a regression model. These are really important to ensure that all of these assumptions are met so that you can be confident in the inference you're going to make when you're running your data. We applied long transformations to normalize atrazine exposure and diabetes rates, and then any outliers were removed. We also did cross-stat validation. So this is the sensitivity analysis I was talking about. We performed a 10-fold cross-validation, which essentially just shows how robust the model is. And it prevents overfitting, because when you overfit your model from a statistical perspective, essentially, it truly creates a lot of noise, and those residual noise distorts or inflates a relationship that isn't there. So we wanted to make sure that we, even at the lowest power possible, if we're still able to detect a significance, that means that there truly is some type of correlation between the two, right? We talked about median imputation, and again, it reduces outliers, it reduces skewedness and distribution. So it was really important that we did that. When we're looking at how we uncover the atrazine diabetes link, we again made sure to account for a lot of the things that can be associated, and it's been well documented in the literature on what increases diabetes incidence. So our final model included, because we had many different things, but our final model included atrazine levels, median income, BMI, obese, because we had different levels of BMI, but there was a lot of different interactions within the model. So it was causing overfitting. So we had to remove the other BMI levels. So obviously BMI obese is the most associated with diabetes outcome. Education level. So those are the three. We didn't have enough data from racial demographics to truly look into the racial aspect. So we did have to remove that. So those are the aspects that we included in the model, as well as fruit and vegetable consumption. So descriptive results, again, this kind of shows, you know, the means, median, minimum, and maximums of all of the different variables that we started out with in the total population. So we had the future fruits and vegetable consumption, physical activity, the atrazine exposure, BMI, whether or not obviously the patient, you know, had diabetes, the age. We started off, like I said, with the ethnicity, but had to then remove it, because as you can see, there's drastic impacts. The other create a really good representative aspect to include. And so that's why we had to remove those. And then we had medium income, and then as well as education. So of course, I'm a geospatial environmental epidemiologist. So of course I had to map this out. So these again is the counties, all 67 counties. This shape is the state of Pennsylvania. And so this is actually labeled now with all the different counties. So the darker the color, the higher the diabetes prevalence. And then the bigger the purple diamond is the higher the atrazine exposure. So as you can see in the darkest parts of Pennsylvania, you have the biggest diamonds. So before we even actually completed the multilinear regression, we were able to kind of see that there is definitely something here. There's a pattern, right? There's a trend that, you know, where the prevalence is lower, the diamond is also lower. So, you know, we were able to just visually see that already. The areas with high prevalence of diabetes has high atrazine exposure. But again, all circumstantial, right? There could be something modifying that effect. So the only true way to know that is to run a regression. And so we did. And our results for that regression showed that it was significant. So there was a significant association between diabetes outcome and atrazine levels. So the p-value was less than 0.0001, which means that we did you know, reject the null hypothesis, which means the alternative is true, and that alternative is that there is an association between atrazine and diabetes. We accounted for obesity, which also was significantly associated, right? We accounted for age, we accounted for income, we accounted for consumption, we accounted for certain ethnicities as well. As you can see, they were not significant when we first did this model. So we did cross-validation, we did bootstrappings, and all the assumptions were met when we ran this regression results. And so with that being said, we were really confident in what we were stating when we said that there is an association between the two. So our conclusion here was that atrazine exposure and obesity are key predictors of diabetes. Obesity, we knew that atrazine exposure is not very well yet known. We need to focus on targeted interventions and start looking at the chemical environment, right? We have the built-in environment, the intra and interpersonal relationships type of environment, but we often forget about the chemical environment, how that's impacting us. And with, chemicals being dumped illegally and policies being lifted when we're thinking about the environment, this is something we have to think about even more apparent. If the chemicals can be in the food we eat, it's definitely in the environments that we live in. And so looking at how that impacts us and how that is actually contributing to our health outcomes is very apparent and should definitely be a part of the One Health approach. When we look at the educational impact, higher education for those who had a bachelor's degree is protected, right? So if you have a higher education, you're more likely, in a perfect world, obviously we're seeing there's some mediating facts to that, depending on, ethnicity, depending on where you live, depending on job opportunities. But overall, if you do have a higher education, you are more likely to live in better neighborhoods and have have access to healthier choices. We're thinking about policy recommendations. We truly need to start thinking about stricter atrazine regulations and not just stating, overall regulations that doesn't necessarily account for temporality, and really focus on how we can truly think about prevention. Future research should look at individual level data. Again, this was aggregate data, so we can't make any individual conclusions from this. So we need to look at the long-term effects truly, and maybe collect blood from patients who have diabetes and who live in high exposure and high SVI areas. And so the key takeaway is that the study highlights the strong link between atrazine and diabetes, and is underscoring the need for public health and policy interventions. So what's next? We have, but since the NIH has been kind of on a hiatus, we did have a grant, no longer, but we did have a grant that we were essentially applying for to create individual-based studies to detect that stronger causal relationship. Despite this, though, we're still working with local practices and working without the grant to make sure that we are continuing the work and working with local organizations. And so potentially, like I said, we could further our partnership with local hospitals and truly try to find those indicators at an individual level. All right. And we have we are published in the Journal of Environmental Ecology. So this study has been published since we've completed our research. And that is everything that I have. So I will open it up to questions.